When we learn about meiosis, we often learn about it in comparison to mitosis, even though these are truly independent processes. Similar, but very different. And so we already know, going through mitosis as our comparative, that the purpose of mitosis is to make identical copies of a cell to produce somatic or body cells such as muscle cells, stomach cells, pretty much almost any cell that's not our sex cells, our gametes. And in mitosis, we start with a diploid cell, right? We start with one diploid cell and we produce two identical cells to that original cell. And so the number of chromosomes in that starting cell compared to the daughter cells are the exact same. We start with 46 chromosomes in humans, that is, and we end with 46. So diploid to diploid. And mitosis is not for the purpose of genetic diversity. The point of mitosis is to have the cells be identical, because the whole point of the replication process here is to ensure that one cell is able to replace a previously existing cell, right? We want those cells to act for growth and for replacement, all right? But in contrast, meiosis, as you all already know, the point is to make non-identical gametic cells, so those sex cells, sperm and egg cells, that are not identical to help promote that genetic diversity. So the types of cells that are produced are gametes only, and we go from one cell, one diploid cell, to in meiosis 1, we go, I'm sorry, you can hear my cat, that's kind of funny. You go from one diploid cell to two haploid cells in meiosis 1, and then from there to 4 haploid cells in meiosis 2. So we start with 46 chromosomes if we're looking at a human cell, and we end with 23 chromosomes in each one of those gametic cells, our sex cells. So those are each haploid cells. And so the point of this really is to promote genetic diversity, right? This is why eventually when you produce offspring, you look different from your siblings as well as from your parents, right? So what I want to focus on here, though, these are things that students are able to grasp decently well. Where students really struggle, though, I find, is right here, where we look at the types of cells produced. Sometimes students get confused as to where those cells come from and how they start and where they end in this meiotic process. So let's explore that really fast. To rate key details pertaining to meiosis in biological males versus biological females. So in biological males, meiosis takes place after puberty, all right? And so where? It takes place in diploid cells that are within the testes of the biological male. So you have diploid cells within the testes that undergo meiosis 1 to produce those initial 2 haploid cells, and then meiosis 2 to eventually produce those final sperm cells in the testes. And this all takes place after puberty. In contrast, though, in females, meiosis begins during the fetal stage, when a series of diploid cells enter meiosis 1. And at the conclusion of meiosis 1, the process comes to a brief pause or a halt, and the cells gather in the ovaries. Then, at puberty, meiosis resumes. One cell at the end of meiosis 1 enters meiosis 2 each month. And the result of meiosis 2 is actually a single egg per cycle, because the other meiotic cells disintegrate, and each egg cell contains 23 chromosomes and is haploid. Make sense? Okay, hopefully this helped clear things up.